Following is a conversation with Dr Indrani Mukhopadhyay, lecturer at the Institute of Medical Sciences, Microbiology and Immunology Group at the University of Aberdeen. As always, I wanted to thank you, the listener, for tuning in and for supporting the podcast. It's fantastic to get the feedback that we've been getting about people enjoying it. And if you are enjoying it and haven't yet liked and subscribed, please do that. Please consider kindly giving a 5-star review because it really, really helps the podcast. In fact, it's the best way to support the podcast. What you can expect to learn from this episode is what is the virome. And very briefly, it's a term used to describe all the viruses that are in the microbiome. And there are a lot. And over the last 10 years, we've started to understand more and more about why they're important in both health and disease. Indrani is an expert in the virome and has been involved in some of the seminal research to characterise, profile, and help us understand why the virome is important. So if you're interested in that, this is a great episode for you. We also spoke about probiotics and what a next generation probiotic might be in terms of its composition, structure, and function. We spoke about Indrani's career progression. She's been all over the world, involved in exciting research, and setting up her own new lab recently here in Aberdeen. So we covered a range of topics. We also had a little debate, which we'll release separately, about which part of the microbiome is the most important, which was fun. And finally, we spoke about inflammatory bowel disease and how the viral elements of the microbiome might be important and how specific depletions of bacteria might be important too. And ideally, we could take those depletions, turn them into a product and give them to patients with IBD to cure their disease or maintain remission. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. How did you get into the field of the microbiome?
Well, it's before I got into the field of microbiome, it was actually entry into gut microbiology. So I started on gut microbiology during my PhD studies. So I worked on a project which was funded by Wellcome Trust to look at molecular epidemiology and correlates of protection against rotavirus infection in a birth cohort of children.
And what's rotavirus?
So rotaviruses cause gastroenteritis in children less than 5 years of age. It's one of the leading cause of gastroenteritis in low and middle income countries now. And that's what we were studying in a birth cohort. And I was doing this in India, in a place called Vellore. And it was in the lab of Dr. Gagandeep Kang. And she's one of the leading virologists in India. She's a trailblazer in her field and great mentor, has been a role model, a big influence in my life, not only mine, a lot of my colleagues, especially women scientists. So that kind of ignited my interest in enteric viruses. And so we were doing this PhD project. We recruited a birth cohort of 452 children, who we actually followed up for around 3 years. We intensively followed up by using quite a large number of field workers. So we collected fecal and serum samples at predetermined time points and also at every episode of diarrhea. And so my role was to characterize the rotaviruses, the genotypes, by sequencing, by PCRs, and kind of characterize what was circulating, what were the different strains that was there in that particular cohort. And because this was a multi-center study, this also gave me the opportunity to visit other labs. So I got the opportunity to train in Baylor College of Medicine for around 6 to 7 months. And this is where I developed these immunoassays from these circulating strains. We made recombinant rotavirus proteins, which we put in these assays to check for immune response against IgG, IgA, serum response. And I developed those assays in Dr. Estes' lab in Baylor. Again, a great...
Where's Baylor?
Baylor is in Texas.
What were some of the differences between academia in Texas, USA versus where you were before?
So in terms of resources, the enthusiasm was the same.
Okay.
But of course, the level of resources that was available at a price that could be affordable drove us there to do those kind of developmental work. So once we developed those assays, of course, I got them back to Vellore and established there and did the actual study back in Vellore. I also actually trained in health protection agency in London in the Enteric virus unit during that time on the various molecular techniques. So got a lot of exposure during that time, worked with a lot of enteric virologists, which kind of like, again, drove me towards this particular field. And for the study itself, one of the key findings from the study was that more than 3 rotavirus infections were actually needed to confer protection in that particular cohort.
Is that right?
Yeah, it was published in New England Journal of Medicine. And
which for the listener is one of the highest impact ones, right?
It is a good journal people kind of like follow. And so it was important for another aspect was that was the year also when rotavirus vaccines were being launched globally for the first time on a 2-dose regime. So our study kind of informed that maybe there may be more doses needed to acquire a better efficacy for these vaccines. And that kind of borne out in the studies which followed. It was seen in the higher income countries, of course, efficacy reached more than 90%, which was excellent. We were fascinated and very happy for it. But again, in the lower income countries, middle income countries, the efficacy didn't reach as much. So it ranged around 50% sort of. So this led to, looking more into it, using more indigenous strains, because the Rotarix vaccine, which was a GSK vaccine, which was initially launched in India, was a G1P8 strain, and it was a monovalent vaccine. It was one of the prevalent strains worldwide, but there were a lot of more heterogeneity in the Indian and other low-income countries, which probably drove that. And therefore, it led to using more indigenous strains to develop more, local vaccines, which were more cost-effective as well.
Mm-hmm. And something you're, I think, elucidating here is that a lot of drug development happens in the Western world. Vaccine development, although it's not a drug, diagnostic development. And we were touching on this before the podcast, actually, weren't we, in terms of ethnicity and potential genomic differences and environmental differences. What you're saying, I think, is that the vaccine was developed in the Western world by GSK.
And Merck. There were two vaccines.
So big pharmaceutical companies who are based in the Western world and whose shareholders are almost all in the Western world, I think. Or at least I'd imagine so. And there was a large difference between the effectiveness of the same vaccine in the context of the same infection that you're trying to prevent based on where the people were being vaccinated.
Yeah, to be fair for the vaccine companies, they did do a trial for one of the vaccines in Latin American country. But I think because fortunately, we were also doing this big cohort study around the same time. So we had a lot of epidemiological data to support and see what was actually going on in the field. So some of the things which we noted, which could have affected the efficacy at that time, were things like malnutrition. Yeah. So comorbidity, a lot of other pathogens in the same person. And many other things like even, crowded living conditions, sharing the same household, open air, like, cooking, and then proximity to domestic animals even. Because this kind of like led to a lot of transmissions, zoonotic transmissions, which we also documented during the study. And we kind of saw like zoonotic strains like G10P11 kind of like causing symptomatic infections in children at that. In those 3 years, in that short span of time, we could document so many different things.
It's fascinating because you could say there's a big push for diversity in clinical trials. You need a diverse population, even within the same indication or disease. You want a range of ethnicities and ages, ideally, and other factors. However, even if the drug companies had tried to account for that, I think what you're saying is it still wouldn't have kind of covered or taken into account all of the factors that may be important, like malnutrition, for example.
Absolutely.
Which could be huge. And
yes.
I guess it kind of makes sense. If you're malnourished, then your immune system's not going to be firing in all cylinders.
Great, yes.
So how then did this essentially allow you to segue into the microbiome? Because at that time, was the microbiome something people talked about?
Not really. Like, we did look into the So we were trying to close the diagnostic gap. So we were collecting diarrheal samples. We did see a lot of viral infections, of course, norovirus, rotaviruses, even cryptosporidia, like those kind of infections were predominant. There was Campylobacters and others, Giardia, which you wouldn't probably find so much here in the Western world. But not much of the concept of microbiome at that stage. We were looking at individuals, right, causative agents, and so it came much more later. So after I finished my PhD, I had the opportunity to come and work as a postdoc here in Aberdeen University in Professor Elomar's lab. So
big name in the microbiome, yeah.
Yes, so Georgina Holt, of course. And that was a time, that was kind of the period where the microbiome was taking off, and I got the opportunity to work in that space at that time.
And when was this, sorry?
It was a decade ago.
10 years ago, yeah.
10 years ago, sort of.
Things were just lifting off. Yeah.
Yes. And interestingly, my first project when I came here was a follow-up project on the rotavirus vaccine study. So incidentally, when I came to Scotland, that was also the time when the rotavirus GSK vaccine was being rolled out in Scotland. And Health Protection Scotland just realized they didn't have any baseline data on rotavirus epidemiology in the whole of Scotland. So they quickly looked for people who, were able to work in that field. And I was kind of contacted. And
for the listener, sorry to interrupt, but for the listener, can you explain why having baseline epidemiological data is important?
So it is important to judge how good the vaccine is, how it has been able to reduce the disease burden or change it. So just to check the efficacy, you need to know what was there before to see how it has changed, the intervention has changed. So
you need to know what normal is.
Yes, right? What normal.
Pre-vaccine.
It was not normal, of course, it's disease. So what were the prevalent strains, basically? So what we did was from all the health boards in Scotland, Edinburgh, Glasgow, Inverness, Perth, Aberdeen, we collected all the rotavirus samples and genotyped before the vaccine. So it was a kind of retrospective study for the pre-vaccine era. Thankfully, they had samples stored in their freezers, which we could access. And then we collected some after the vaccine, and we did a very neat little comparison to show what was happening in the field. And we saw that the predominant strain, which was G1P8, which was also the vaccine strain, almost completely gone in the post-vaccine era. It dropped from almost 80% to 20%. And also the vaccination, after the vaccination, the hospitalization rates were down by 74%, which was great.
So the vaccines are working?
The vaccines were working, and the uptake was very good, above 90%. So it was a win-win for everybody. And so we followed up for several seasons to see that that good effect still continued and the vaccines were still efficacious.
So in the context of a vaccine and seasons, do you expect there to be some sort of seasonal change in the specific strains? Does that happen with all viruses or only some viruses?
All viruses do evolve. So rotavirus is an RNA virus. And so all viruses, it's actually a double-stranded virus, and it is segmented as well.
What does that mean? Sorry.
So this is a rotavirus is more complex. So when you talk of viruses, they evolve. Of course, especially the RNA viruses, they have a very faulty replication system. They don't have the checks and balances. So every time they replicate, they put in an error in their code. And therefore, that's how the mutations build up. So slowly they change.
Is it an error or is it some sort of intentional evolutionary?
Probably it is error prone. It is just error prone. They don't think so much.
Right.
It's just error prone. They're very fast evolving. They're error prone. They're very simple. They don't have these complex systems for checks and balances. So every time they replicate, they just put in some errors, and that's how they evolve, and that's how slowly they drift. So that is called the antigenic drift in the evolution. So in the rotavirus, there is another layer of complexity because they are not a continuous segment of RNA, they are segmented. So they have these 11 segments which codes for 5 to 6 different structural proteins and another 5 non-structural proteins. So when you have a condition where 2 different rotaviruses meet in one person, so suppose one person has acquired a human one, another from a cow or from some other cattle, they can actually exchange segments and then they can evolve into a very different virus very quickly through reassortment. So that's when you say it's got an antigenic drift. So it like antigenic shift, So it kind of like changes into a different virus altogether. So it does evolve, but we haven't seen it like the seasonal flu. So that one like probably changes more every year and you need a different vaccine. But for the rotaviruses, we have seen introductions, zoonotic introductions, G9P8, G10P11.
Zoonotic means it's come
animals,
from an animal.
From an animal. So we have seen those kind of things, but the dominant strains sort of remain the human 3-4 strain, G1P8, G2P4, G3P8, G4P8. Those are the dominant 4 strains which has kind of stayed consistent. And even after vaccination, we haven't seen too much changing.
Okay. Why is it that the seasonal flu is so changeable?
It has to do with the RNA virus it's the nature of the virus, as I said. They just change very fast, mutate fast. So it's a smaller virus as well, quick change.
Right.
And therefore, there's the need for every season updating based on what's circulating and what's evolved. And for any viral infections, therefore, I feel prevention is always better than cure as well.
Okay, so by prevention, do you mean vaccine or do you mean washing your hands?
Well, washing your hands do work. They do work in checking transmission to a certain extent, and that's a very basic thing, probably advocated for hygiene purposes. Yeah. And also the vaccines.
Yeah.
So the vaccines, I would say, because there are so few antivirals, you would know, like being a medic, like how many you can actually count them in your hand. Probably like most of them are against HIV, and of course it's revolutionized after the HCV, HPV. Yes, very, very few which actually were, and then they again come up with resistance. Like a lot of resistance also happens. So
the HIV ones are awesome.
Yes,
they're really good.
of course, but it was not always like that.
Indeed.
It started off like gathering a lot of resistance,
took a long time.
Then people learned like how to combine therapy, heart therapy, and so it is now, of course, just a tablet you take, which can keep you going. Yeah.
Some people listening might be going, well, why is there not antiviral for the cold?
For the cold?
Yeah, the common cold.
Yes, because it's so varied. Like it's not one particular species, not one particular strain. They change so much, so therefore it's difficult to target.
And there's not some universal way of killing all viruses and you could just spray it up your nose?
Not that we have found on, so that's a holy grail. At least for the common cold.
A pan-antiviral
that
therapy.
Would be really nice.
For all variants ever of the cold.
Yes.
Is it worth just touching on how diverse viruses are in terms of the genomic elements that make them up? Like,
oh yes, of course.
Huge diversity, right?
It is huge. So there's a person called Baltimore who also got a Nobel Prize, but not for classification of viruses, but for reverse transcriptase. Anyway, so he has this classification system based on the nucleic acids, and there can be more than 5 or 6 types. So you have, of course, the DNA viruses.
Yep.
In that DNA viruses, you can have single-stranded DNA, you can have double-stranded DNA, and then there are variations. And then there are RNA viruses where you can again have mostly single-stranded, but plus sense, minus sense, and also double-stranded. So there's a lot of complexity in the viral world.
Yeah, and just one or two changes to that strand of RNA or DNA could produce
make them very different.
Very, very different. So that's why it's difficult. And what about the viruses in your gut?
Oh, that's of course very fascinating as well. So viruses are everywhere, as you know, like we're eating, breathing.
Yeah.
Are
they really everywhere?
They are everywhere actually, and they are in the range of 10 to the 31 viral particles on Earth, and most of the
10 to the 31.
Yes.
Okay, that is a big number.
That is a very big number.
I think the number of stars in the Milky Way is like 1 times 10 to the 10 or 11.
That's the analogy people usually give when they try to count viruses. But they are the largest, you can't even call them living, but they are the largest entities you can find. And most of the Earth's viruses actually come from the oceans. But of course, the humans also has a large number of viruses, and collectively we call that the virome.
Within the body.
Within the body, human. So again, the various parts of the body can have different sites have different viromes of their own. For example, the skin can have their own virome. The kidney can have its own virome. And of course, the one which fascinates me and one which has the largest number of virome is the gut. So that has trillions of microbes, and one of them is, of course, the viruses as well.
And just before we delve into that, because we're going to talk about that for a while, just wanted to make the connection for the listener between your original rotavirus research and what we're talking about now, which is the virome. So rotavirus is a enteric virus, which means that they essentially infect the GI tract.
Yes.
So the enteric element means it's gut.
Gut. It is a gut virus.
It's an enteric virus. That means it attaches to the enteric system, the intestinal tract, yeah?
Yeah, so when we talk about gut viruses, there can be different kinds. Mm-hmm. So one, of course, is the eukaryotic viruses, and rotavirus is one of those eukaryotic viruses. So these are viruses which can actually infect the colonocytes, enterocytes actually, in the upper gut. So apart from rotavirus, there are other viruses like norovirus, some strains of adenovirus, other circoviruses, aniloviruses. There are lots of eukaryotic viruses, and usually those are the ones which we have been studying for a very long time. These are the ones which we knew of because they are pathogens and there were means to study them. So these are the ones which we have been traditionally looking at and studying for a very long time. And there was a lot of unknowns in this gut virome. Known unknowns. We knew there are other things, but we didn't have the means to study them.
10 years ago.
Yes, 10 years ago or so. So with the advent of metagenomics, it gave us the tool.
Metagenomics?
So it's a sequencing technique where you can actually chop off pieces of DNA from any environment and can read the DNA sequences and then match it up with a database to identify what's there.
And can that sample be taken from anywhere?
Yes, it can be environmental, it can be from your gut, it can be from your skin.
How transformational has metagenomics been for the field of the microbiome?
It has been a huge transformational effect because a lot of these microbiome, be it the virome or the bacterial side of things, a lot of them are not easily culturable. So for the first time by metagenomic sequencing, 16S RNA sequencing, we got a sense of who they are, what is there.
Right.
So, and then of course we could use those information to design better culturing tools. Right. And now we have much better ways to actually match up the genotype with the functional sites by actually phenotypically studying them.
So if I can take you back to when you just started to get interested in the virome, what was known about the virome pre-metagenomics? Like, what was the state of the field like?
Yeah, as I said, there was very little which was known regarding the bacterial viruses. So mostly it was the eukaryotic viruses which was being studied because they were so important. They were the pathogens. And then we have the gut virome actually is predominantly filled of bacteriophages. So this bacteriophages are viruses which actually infect bacteria. They don't infect humans or other hosts. They specifically target the bacteria. So these are the kind of like viruses, bacteriophages, which are predominant within the human gut. And these are the ones which like we didn't have the means to study before. So one of the main drawbacks of studying any kind of viruses was we didn't have a marker gene. For example, in bacteria, we have the 16S RNA gene. So that is used to, identify for the fungi. We have the 18S, the ITS. But for viruses, we don't have that kind of a marker gene. And therefore, this metagenomic sequences was the first time that we could actually study them in great detail. And it showed us that not only the eukaryotic viruses are there, we also have this huge population of bacteriophages. And there's another kind of viruses which are also present in our gut, which is the plant viruses. It comes in very transiently through our food, our diet, and they are not known to infect humans so far, but they are present in a lot of studies, and mostly they are RNA viruses.
Now, these bacteriophages, they must be very small if they're infecting bacteria.
Yes.
Right?
Yes.
Are they, so they're very small and they only infect bacteria? Is that right? They don't. So what happens?
They're very specific. They specifically infect bacteria of only one particular type. They can be even strain specific.
Wow.
Yes. So they are very important probably in the gut ecosystem. And they drive a lot of things as well because they are in a predator-prey relationship with the bacteria. They could be modulating the bacteria as well. So it's not only like they are targeting the bacteria, they're preying on them, lysing them, destroying them. They also have another important characteristics, which is like they can integrate within the chromosome of the bacteria.
The virus can integrate?
Yes.
Wow.
So what they do, the bacteriophages can replicate in various ways. One is the lytic phase, which I just said. They infect a bacteria, replicate themselves, and destroy the cell, come out. They can also go like...
Sorry, just for the listener. This lytic phase is really interesting, isn't it?
Yes.
Because you can look at a photo of a bacterial cell which is being attacked by bacteriophage, and it's like something out of a sci-fi film, isn't it?
Yes.
They're like little spaceships
landing
because they've got these,
injecting the DNA inside.
They've got these legs which kind of come out, and then you've got the middle bit which is the
injecting the DNA. Yes.
And then what happens? How does it explode? Like what happens to the bacteria?
So they have endolysins, which like enzymes, which like disintegrate the bacteria once their progeny is formed. Yep. And also there is, as I was saying, the more interesting bit was the lysogenic phase where they actually integrate within the bacterial chromosome.
Yep.
And this kind of gives a different functionality to the bacteria as well, because they can bring in genes which were not present before. And this could lead to either the bacteria becoming more fitter or more virulent. So that way they have modulatory effect, like not only by killing them, but also by changing them, evolving them. And this is how a lot of AMR resistance is also transferred into a bacterial cell.
Right? So the phage?
Yes, the phage carries a lot of these AMR genes.
Every time I do one of these podcasts, it just gets more and more complicated. It's not getting any easier.
Yes.
So they're both friend and foe.
Yeah.
The best way to describe them as a foe, I guess, is imagine a large number of mosquitoes landing on your body. The mosquitoes inject something and then you blow up.
Yeah. Thankfully, the mosquitoes don't do that.
Thankfully, no. Thankfully, they're not as adept as the bacteriophage. So that's one. And then the other is actually in contrast to being a destructive force for the bacteria in your gut. They can also help the various different bacteria level up. So for just to give a random example, maybe they could confer a gene which made them more resistant to an antibiotic this person was taking a lot of for some sort of chronic infection or prevention or whatever. And the bacteria that's then got that sequence that confers a resistance has leveled up and it doesn't get killed by the bacteria. Is that a good way of describing it?
Yeah.
And taking us back to then when you first started to get interested in it, there was a lot not known. But did we have some theories about the bacteriophage, that they must be there? How did we know they existed and that kind of thing?
Yeah, the first kind of studies were very basic. So even before metagenomics, it was just transmission electron microscopy. So you just looked at them under a microscope and you could see these phages. And they have very different structures as well. You can classify them based on whether they have tails, whether they don't have tails, and the size of their tails. So those were the kind of things, and that's how it started, by transmission electron microscopy, and then it was followed up by metagenomic sequencing, where we found that, of course, there's one particular class of phages which is overwhelmingly present,
right.
Which belongs to the cordoviruses family. And you have several of these cordoviruses, like Cyphoviridae, Myoviridae, Podoviridae, Microviridae. So those very different kinds of viruses which are present in the gut.
Do you not think it's just so unbelievably cool that within each of our guts, 24/7, 365, there is this super dynamic interplay between billions of phage and billions of bacteria, and we're just unaware?
Absolutely, and as you just mentioned, they're in the region of 10 power 8 per gram of intestinal material, VLPs, virus-like particles, which is very similar to slightly lower than the bacterial content. So it's a dynamic situation. They are also in a predator-prey relationship. So they are just not static and just doing nothing to each other. They are interacting with each other in a trans-kingdom manner all the time.
Oh.
And that, if you take into the host genetics into play, can lead to different disease phenotypes.
Now, a lot of our research has been very bacterial-centric. You mentioned interkingdom. Can you just talk a little bit more about what you meant by interkingdom and why it might be important?
The trans-kingdom interactions, as I said, they're microbe within our gut or any part of the body. They don't exist on their own. They're constantly in touch with the various other microbial components. For example, if you take the viruses, they're in constant, relationship with the bacteria, also the fungi, the protists, everything together. And you also have the host genetics and the host immune system. So everything together establishes that gut ecosystem and who would survive in that particular niche.
Why has our knowledge of the bacterial side grown so much faster than the viral side? Something to do with the marker genes, maybe?
Yeah, probably because it was, first thing is it's much bigger than a virus. So we could see them under a normal light microscope. We didn't need an electron microscope so more access to that. Of course, their presence was known for a very long time, but we didn't have the tools to actually study them till very recently.
i.e. metagenomics.
Yes.
So what have we learned then about the virome over the last 10 years?
So what we have learned was that in various disease conditions, just very similar to what we have seen in the bacterial side of things, there is an alteration. So there is an expansion actually of one particular group of viruses called cordovirals in certain disease indications like IBD. And again, we haven't been able to decide cause and effect. So we don't know, chicken and egg situation still arrives. So a lot of research is now going into understanding who's driving this change. So now we have got a better understanding of who are the players who are present. So we are more into geared up for mechanistic studies. We need to really tier that properly and understand who's there and how they are now changing the environment, how they're interacting, and how they're bringing about the dysregulations that we see.
And are you actively involved right now in virome research, or are you focusing your efforts on other parts of the microbiome at this point in time?
One of the parts that one of the research stream that is I'm quite interested in is looking at the gut viruses and what role, immunomodulatory role they have in various immune dysregulations. And one of them is, of course, inflammatory bowel disease.
It could be huge if they actually had a key role to play in developing these inflammatory symptoms, right?
Yeah, because now from the bacterial side of things, it is quite established for the last 2 decades almost. We know that there is alteration, there is change in the bacterial component. There is more of harmful bacteria, more proteobacteria, less of the beneficial bacteria. But we don't know yet what cause. It's, of course, a multifactorial disease. But we still don't know what's the trigger. What brings about that change? There has to be, it's almost like a two-hit theory. So you have this change happening, but on top of that, something else happens which drives them towards this irreversible, immune dysregulation, this aberrant immune response. So and my hypothesis is that the gut viruses could be the driving force in that.
Now, if someone was to say to you, Indrani, what's the most important part of the microbiome? What would you say?
Well, I wouldn't say one is more important than the other. I think they all coexist together and they have their own functionalities and they drive the dynamics. So a healthy microbiome should have like all components and nothing should be missing. Of course, not the pathogens.
But you couldn't make the case, could you, for the viruses being the most important?
Not yet.
Not yet, no.
Yes.
Because that could be a fun debate to have.
Yes.
I could try and make the case for the bacteria.
Yes.
So what is actually known as?
So I might make a little bit of since you
let's have a debate.
Talked about a debate.
Let's have a debate. Yeah.
So I see the.
So the question is, what is the most important part of the microbiome?
Who's driving the changes?
Who's in charge?
Who's in charge?
Yeah.
So as I mentioned, these bacteriophages, they not only have a direct effect on the modulation by just killing them, they also can modulate by changing their phenotype, by changing, their characteristics by inserting genes. So that's one way. So you see that they are the drivers of changes in the bacteria. And then there is a newer thread of study, like research teams coming up, which actually shows these phages can actually, without the mediation of bacteria, can directly talk to eukaryotic cells as well.
What? How? That seems not possible based on everything.
Yes. So now newer research is indicating that there might be interactions which are possible. So when there is a breach in the gut lumen, and when the epithelial barrier is breached, and when there is this alteration in the bacterial and the viral component, so when there is an increase or expansion of these viral components, what happens is you'll have more killing of the bacteria.
Right.
So when more killing of the bacteria happens, more these bacterial parts, which are like PAMPs, so pathogen molecular entities, which will be like then targeted and infiltrating into the epithelial cells. And this could lead to a lot of immune, activities leading to granuloma formation,
So that's interesting, but
So the other thing is, so they have been now found in various sites within the body. So usually
phage.
Yes, bacteriophages.
Beyond the gut.
Yes, beyond the gut, they have been found in blood and other sites. So.
But I thought blood was sterile. It's not sterile? I'm joking, I'm winding you up. I know it's not sterile, but the listener might know it's not sterile.
So the thing is, like these phages, as I said, they stay as prophages within our gut, so which means they are within the bacterial chromosome. But when the bacteria lyses, they can be coming out and, like having interactions with the immune systems themselves.
Right.
And also now we know that they are actually lining the mucus layer of the gut epithelium as well. So they might be having a bigger role in immunomodulation, immune training, immune tolerance, very similar to the role of the bacteria.
So you're making a very good case for why
they are therefore known as the apex predators.
Well, it's my job to push back a little bit. Now, you've talked a lot about bacteriophage.
Yeah.
We've not talked much about the enteric viruses, and you've spoken a lot about why you've gone a bit further and gone why they're important for both the bacteria and the human. But if I was to just sort of push back on you a little bit, because this is a debate, these viruses, the eukaryotic viruses, the human viruses, they just seem to be really pesky, and all they do is either do nothing or infect us. And also, none of the viruses are metabolically active.
The human viruses, the eukaryotic viruses.
None of the viruses you've described produce the metabolites which benefit the host, whereas the bacteria do.
Yeah.
So, given that they don't produce any metabolites, can we really make the case to say that they're the most beneficial?
They are, of course, not in the sense as how bacteria are benefiting us, but they are benefiting us by probably modulating the bacteria for us. So they keep in check that one population of bacteria doesn't overgrow. So there are various epidemiological theories of how they do that. I'm not going into the details of that, but they probably modulate the bacterial population and shaping them and keeping them healthy.
And do the bacteria not do that themselves by competing for nutrients and bacteriocins, the peptides they produce, which are like sniper rifle?
They within themselves, they do that as well. But at the transkingdom level, I think the phages do that as well.
And they're maybe more important. Okay, what about the fungus, though? So the fungus are the biggest cells.
Yeah.
And they also, like the phage and viruses, have been rather understudied. Mm-hmm. But they're definitely there. They
are there, and I'm sure they have an important role to play as well. I haven't done much research into that area.
Yeah, the fungus has been neglected. Yeah, yeah. Right, so we can conclude that part of the debate then. You've made a very strong case for why the viruses are the
definitely
most important.
needs to be looked into more details. We are scratching the very top layer, and so we need more understanding.
For the listener that's tuned in, I tried to make the case to say that the bacteria are metabolically active and they produce a lot of metabolites. Short-chain fatty acids are the most common example, but they do produce metabolites from other bodily produced substances, like, for example, bile acids. They're heavily involved in the conversion of bile acids from primary to secondary, and the secondary bile acids are important for suppressing other bacteria and doing all sorts of important things. So I think what we've concluded is the viruses do a lot. They keep the bacteria in check. They can enhance the fitness of a bacteria through injecting some genetic material into it. And what you're describing at the end of your pitch for why the viruses are most important was fascinating, because it was talking about how they can modulate other parts of the body and are involved in immune system training and tolerance and things like that. So good job. You've done your viral colleagues proud. So finishing off that part of the debate then and bringing you back to your research. Don't worry. We got time, don't worry. What are you most excited about at the moment within the microbiome field?
I think the more mechanistic studies at the moment will be key in understanding disease pathogenesis. And that's what excites me, where we can actually isolate a particular, it's just not genomics. We can actually have a strain and put it into an in vitro, in vivo system and actually study disease mechanism.
And has that been lacking up until now?
That has been quite because of the state where we couldn't culture a lot of these bacteria, strict gut anaerobic bacteria, or for that matter, even bacteriophages. So all of these, because they have the anaerobic host, so you should be able to grow the host first in order to be able to grow the viruses.
Right.
So all of that is based
I see what you're saying.
Yes, it's all based on this gut anaerobic microbiology.
Hard.
And not many labs are geared to do that. And
so can I just, I don't understand myself. So I think what you're saying is, if you want to study the anaerobic bacteria, that's one thing, because you study the bacteria themselves. But what you're saying is, if you want to study the phage, they infect bacteria, and if you want to grow the phage, you need bacteria for them to infect.
Yes. So, for instance, one of the most predominant gut phage, the crass phage, the host is Bacterioides. So
Right.
you have to be able to grow these Bacterioides, and only then you'll be able to isolate the phages from there. So they need a host system. And so therefore, this is kind of a very good time to be a microbiologist, virologist, where you have all this little bit of knowledge, little advanced knowledge in how to grow them and how to do this mechanistic studies. And the other important change which has happened is this omics. And
What's an omics?
so various metabolomics, all these kind of genomic studies,
Proteomics,
metatranscriptomics, yes, everything together gives you a more holistic understanding, because what I feel from my perspective is that all these kind of complex disease questions can't be answered by only from a microbiological point of view. So you need an immunologist, you need an epidemiologist, and you need a big data crunching data scientist. And all these things, all these collaborations should come together to answer these bigger questions. And so that's what excites me to in this particular field at the moment.
And we spoke a little bit about women in science.
Yeah.
And something you're passionate about. Why is it a good time to be a woman in science and also someone who's moving into the microbiome field? So I'm kind of pitching that question to the listeners who might be at school or who might be at university, thinking about studying microbiome or microbiology or immunology, or currently studying, thinking about what do I do next?
It is a fascinating field. And I wouldn't want it to be just for women in science. For in general, every student who would want to come into this field, probably this is the right time, because there are a lot of new inventions, a lot of new tools, a lot of new techniques which are available, and it opens up new possibilities. And therefore, there is a lot of chances for doing actually some good therapeutic treatments.
To help patients.
To
help the population. Now, you've just set up your own lab.
Yes.
So congrats.
Thank you.
Very excited for you.
Thanks.
What are the aims of your lab, and what are you trying to achieve through your research?
So one of the themes that I am currently working on is to look at immunomodulatory effect of these gut viruses. And these have been so far again studied in a very disease-specific manner. So I would like to think there might be an underlying similar mechanism by which it changes in a lot of these indications where you see aberrant immune response in, inflammatory disorders. Mm-hmm. So it's not only inflammatory bowel disease. Another area which I'm very much interested is NAFLD, a non-alcoholic fatty liver disease. Mm-hmm. Of course, it might sound strange how gut viruses is even going to affect liver, which is, again, a lot of people think is sterile. So there is
it's not sterile. The liver is not sterile.
Exactly. So it is like you have the portal vein, which supplies all of whatever changes happens in the gut actually can affect whatever is happening in the liver as well. So these mechanisms...
For the listener, 70% of the blood that goes to the liver comes from the gut through that vein, and it's the only vein in the body where blood goes from one organ to another in an oxygenated fashion.
And it also has this most difficult task of dealing with all that toxins which come
from the microbes in your gut.
From the gut.
Yep.
And so going back many centuries ago, probably, like people have said, like a great physicist, Hippocrates has said, all disease starts in the gut. Yeah. So I keep that in mind.
All disease starts in the gut. Yes, exactly.
Yes. So it again depends on who you're speaking to. If you're talking to a neuroscientist, they will say the brain controls the gut.
Yeah, but they're wrong.
So probably crosstalk between the gut and the brain probably is it. But yeah, so those are some of the themes that I'm looking at. And I'm also involved in research in next generation probiotics.
Yeah. So we've talked a lot about probiotics on this podcast. We've not spoken a lot about next gen probiotics. So help the listener understand what's like a classical probiotic and what you're trying to achieve through your next gen research. And when can I take them?
Probably very soon. So
good.
Probiotics, by definition, are life microorganisms which, when taken in adequate quantity, confers some sort of a health benefit to the person or the host. And classically, for decades, what has been used as probiotics are very limited from a very narrow pool of microorganisms. It belongs to the genera of Bifidobacterium or Lactobacillus. And these have been sourced from mainly yogurts or fermented foods or so far. And they've been like more anecdotally used. There hasn't been huge trials which kind of validate their use so far. So there have been indications that they are beneficial, but you didn't have to really prove them so far that they are really beneficial to be used because they are kind of administered in the food route, in the supplements route, not as a therapeutic. But very recently, things have changed. First of all, as I was mentioning, we have more awareness of what's in our gut, and we have the ability to grow them now, these anaerobic bacteria, and we know how beneficial they can be in terms of various metabolite productions, various other activities that it can do. It can have immunomodulatory properties as well. So based on what your disease indication you want to target, you can have a variety of different gut bacteria which could be beneficial in that particular instant. And now we are in a position to actually isolate them, characterize them both genotypically, phenotypically, and do clinical studies to actually prove that they have some benefit. And that's one of the criteria. So therefore, they are kind of called next generation probiotics, which is very similar to actually live biotherapeutics. So people are actually also debating whether they should be even termed as next generation probiotics, because there is no route to market anymore for that. You cannot have anything going into the market which is not already being sold as a probiotics until and unless you have done a randomized clinical trial to prove that it has some kind of benefit.
And if you do that, then you might as well commercialize it as a drug.
Exactly.
Because you can charge much more and you can get payers involved and you can prove that the benefit has some sort of societal cost reduction or productivity enhancing effect or so on and so forth. So now, you're an expert in probiotics and gut health. Are you taking any probiotics yourself on a day-to-day basis?
The answer is actually no.
Okay. Why is that?
So I don't believe that one size fits all really works in this particular field, which has been advocated so far. Take a lactobacilli and it will end all your problems. It doesn't work like that. And moreover, everybody, I don't feel, needs to be actually taking a probiotic on a daily basis. So there are certain needs when your microbiota is perturbed, maybe when you have taken a course of antibiotics, or where you have lost, to replenish something which is not there, some species, keystone species is lost, and you cannot get it through diet. Those are the kind of instances where I would advocate taking a probiotic. And another instance is in the age spectrum, maybe later, like in the age above 65 or below, even for the children, if there are indications that they don't have a proper microbiome. Probably those are the areas for intervention, and not for every healthy people to be just advocating taking supplements or probiotics. Probably a good diet full of fruits and vegetables and
fibers and nuts and seeds.
Yeah.
Now, if I said to you, I have a machine, special machine, it's called the super probiotic machine, and I'm able to take any strain of bacteria, keep it alive, turn it into dried powder, put it into a capsule, and I can create as much of it as you want. It's a very special machine, which I might actually be working on, by the way.
Fantastic.
Just FYI. Which bacteria would you say you would like me to manufacture and distribute to patients, and why?
Again, it will depend on what's missing in them, like for the disease indication.
But do we know what's missing?
For some, there are indications for, because I'm interested in IBD and like some metabolic diseases. We know that there is a change, alteration in Faecalibacteria, Prosnotzi, and also Roseburia intestinalis, which these two bacteria are my favorites. They're high butyrate producers. They have lots of benefits for colon health. And if you had that super machine, I would ask to make probably Roseburia and F-Pro.
What about Akkermansia mucophilia?
Oh, the jury is kind of out on that.
Really?
Yes, because it also is a mucin degrader. I am a bit cautious when I go introducing a mucin degrader in the system, not knowing how it's going to have an effect.
Can we have a little debate about the benefits of single strain versus consortia? Because there's some people who say, look, single strain is just never going to work. It's just not going to work. So you can take as much Faecalibacterium presence as you want, James, but it's not going to work. And the argument for that is, well, if it was going to work, then we'd be getting sick all the time because single strains of bacteria would become dominant all the time. Does that make sense?
Yeah.
Whereas what actually happens is single strain finds its way in, boom, it gets booted out by the residents who say, see you later. Or your immune system goes, no, thank you. So single strain consortia, what do you think?
I think it would get a better chance with a consortia and also as a symbiotic, I would think, like where you actually pair it with some whatever prebiotic that it loves to feed on. You cannot just push in a bacteria inside your gut and not give it any means to thrive on. So if a person is not taking resistant starch and you're pushing Ruminococcus bromei in, thinking, oh, this is going to do great, it's not going to survive because it doesn't have anything to survive on.
Is there a probiotic which is universally consumed or chewed? Well, we need to talk about what a prebiotic is, actually. Sorry, what's a prebiotic for the listener?
So these are food which are beneficial for the gut bacteria.
Yeah, and they're fibers?
They're fibers, resistant starches.
So complex carbohydrates.
Yes.
Yeah. Now, bring us back. So is there a fiber or a prebiotic which all bacteria chew and I could put it in my capsules produced by my machine, or would you need a range of fibers to support your consortia?
Yes, it depends on your consortia, but for instance, Roseburia wouldn't want, they would like probably inulin better, fructooligosaccharides better, whereas if you have Ruminococcus, it would like starch, resistant starch.
Got it. Okay. So what we're saying is,
depending on how big their carbohydrate active enzyme repertoire is.
Now, there's one company developing FPRO for patients with Crohn's disease. Why do we not have an FPRO probiotic already?
It's difficult to grow. It's very, very fastidious to grow. It is autotrophic to so many different vitamins. It's very difficult to grow.
It's a pain.
Yeah.
It's a pain in the backside.
It's one of the most difficult bacteria to grow in the anaerobic field.
And why have we not managed to, with all the collective brain power of people like you?
We do. It's not that we can't grow it. We do have the means to grow it. We do have lots of isolates in the Rowit. Yeah. From FPRO.
Interesting. And is it hard then to scale its production, or?
Yes, they're very strict anaerobes. So slight, exposure to oxygen.
Done.
Roseburia, FPRO.
They're finished. They're finished. Because I was thinking about starting another company with my super probiotic machine. And I don't actually have a machine, but I think I've got, within the work that I've been doing over the last 6 years, a different way of doing things, which makes my chance of success better. So I just thought I'd ask you that question, and it was very interesting.
So there are spore formers as well. So a lot of firmicutes within the gut are spore formers. So
and they're also important?
Like Roseburia can form spores. So Ruminococcus can form spores, and therefore that could be a mode by how you deliver those as a probiotic.
So for the listener, there's this mixture of spores and non-spores in the gut. Do we have any view of which is more important?
Which group of bacteria is more important? Metabolically, probably the spore formers, the firmicutes, has a lot of
interesting
good use to the host.
Because there's companies developing spore formers only.
Yes.
And you probably know that, right?
Yes.
Excellent. So we've touched on this very wide-ranging and fascinating conversation. We've talked about the virome. We've talked about next-gen probiotics. We've talked a little bit about AMR. We've talked about enteric viruses. We've talked about IBD. We've talked about women in science. We've talked about your lab. We've talked about a lot. Is there anything else you would like to share with the listeners about the microbiome field, gut health, your research, just before we kind of say our concluding remarks?
I think we have covered mostly what I wanted to say.
I'm glad.
Yeah.
I'm good. That means I've succeeded in my role of host, which is an honour.
Yeah.
I've had a lot of fun.
Thanks very much for having me.
Thank you.
It was a pleasure speaking to you.
Yes, thank you. And it's nice to meet in person after goodness knows how many years.
Almost.
And we had a successful collaboration, which was really nice. And you did a stellar job for us. So thanks very much for that. I really appreciate it.
And I would like to continue our collaboration in whatever aspects in the future.
We would like to collaborate too, Indrani. So thank you for that.
Thank you.
And really appreciate you taking the time to come in. It's been a pleasure. Cheers.
Thanks.